Hi, I'm CELINE HANZEN and I'm a postdoctoral fellow at the University of KwaZulu-Natal, where I focus on the migratory and spatial ecology of freshwater eels in South Africa and more specifically in KZN. Here I am going to present the results gathered in the last 5 years on their distribution and decline. Freshwater or anguillid eels are present all along the western Indian Ocean region from Cape Town up to Somalia. They are a long-distance migratory fish with a complex and long life cycle. They are catadromous, which means they are born at sea but spend most of their life in freshwater. They are born somewhere in the Mascarene Plateau of Madagascar, though some of them could be coming from much further away through the equatorial current. They then move to the coast through oceanic currents and migrate up the rivers. The key here is that they depend on ecological connectivity at different levels, between the sea and freshwater, but also at catchment scale. In South Africa, we do have 4 eel species out of 16 in the world, so we do have a pretty good diversity for these species. Here they are their western and southern limit, and the interesting thing is that they only occur in symmetry in KZN. So we do have the longfin eel Anguilla mossambica, shortfin eel bicolor, African mottle eel bengalensis, and the giant mottle eel marmorata. Unfortunately, 3 of these species are now listed as near threatened due to habitat loss and exploitation pressure. Anguilla mossambica, or the longfin eel, which is endemic to Africa, got upgraded from least concern to near threatened last year on the basis of their dramatic distribution decline. Since 2015, we have been focusing on their spatial ecology and particularly looking at their distribution at different geographical scales. So we started collecting current and historical data in South Africa, in KZN, anywhere from any source available. We also sample intensively in KZN through the River Health Program and RAMP for years, seasonally, and more intensively as well, and at catchment scale in the Kashkama and Umgeni. In these 5 years, we have used a range of methods across a large range of landscape, as you can see on the photo. Going from flag netting to electroshocking. So as our 4 eel species occur in symmetry, they do need to somehow share the space. So we looked at longitudinal distribution, and these are the results for South Africa across all data collected, even historical data. So the 4 eel species have significantly different longitudinal and altitudinal distribution. As we can see on the graph, in Mossambica and in bengalensis will go far and up, while at bicolor and marmorata will be normally found closer to the sea and in lower altitude area. Our champion species is Anguilla mossambica with a maximum altitude of 1,700 meters for about 2,000 kilometers away from the sea. So that's quite a trek, and I think it's easy to imagine how distribution can suffer if ecological connectivity is impaired, especially for Mossambica. So we looked at distribution across time in KwaZulu-Natal, and I'm going to show you the result for 2 species. First, Anguilla mossambica, which is the most abundant species. For the species, we also have really good historical data. So here in pink, we have mapped the distribution of the species based on all data collected from the end of the 19th century. So this is basically how the distribution looked like in an ideal world. Now, if we go back 30 years from now, we start to see a shrinking of their range. If we keep going and look in the last 5 years, from when we started to monitor KZN rivers, we do see an even more dramatic decline in their range. Now on to Anguilla bengalensis. The species is at the end of this distribution in KZN. So you can see it was not as widespread as Anguilla mossambica, but it was still present in most of the catchment. If we look last 30 years, we start as well seeing a distribution shrinking. And in the last 5 years, the species has only been recording in only 2 catchment in KZN, the Tugela and the Mfolosi. So if we put all of them together and look at area of occurrence, we can see first that the species always had different distribution in terms of area occupied, which makes sense because, as we said before, bicolor and marmorata would only stay along the coast. They will not go far inland, which means they would occupy a smaller area in any case. But if we look at difference in occurrence at different time frame, we can see that if we compare the last 5 years to the entire data set, we start observing decline. And it's quite dramatic, and it goes over 80% of decline in distribution for M. bengalensis, and 50% for mossambica, which I remind you is endemic to Africa. For the species, the results have actually been used by IUCN among other data sets, so not just for KZN, to upgrade mossambica from least concern to near threatened. So this is not good for our African eel species, but we also wanted to find some explanation, and for that we just thought to look at what was happening at catchment scale. So we did sample to catchment back in 2018 during one self-survey in each, in the Kaiskama and Eastern Cape, which was chosen to be a really good free-flowing system with good historical eel data, and Nyamgeni, on the other hand, tells a very different story, as it is heavily regulated and in general poor state. We also had quite good historical data of eel in that catchment. So as you can see on the photo, we've got the Kaiskama on the left. It's quite pretty, almost pristine estuary with a little village, not much impact as the eye can tell. And then you look into the Nyamgeni that goes through to Durban and through industrial area and heavily dense human population. Estuaries are obviously quite important when it comes to distribution in freshwater, because that's where the baby eels are actually going to come in. So no surprise at all for similar sampling effort. We only caught 5 eels in Nyamgeni against 21 in the Kaiskama. So in summary, we found out that both mossambica and marmorata were present in the 2 catchment. This was expected. We could have also seen bicolor and bengalensis in Nyamgeni, but we did not. We observed differences in abundance between catchment and also between species, with very little eels caught in Nyamgeni, and marmorata only caught in one side in Kaiskama and one side in Nyamgeni. We also observed differences in longitudinal distribution, with only mossambica found upstream of them. Marmorata was always found below first obstacle in the catchment. We performed some more analysis using generalized linear models, and we found out that connectivity, habitat, and water quality were drivers of Anguilla mossambica presence. We did not do such analysis for marmorata, as we caught very little individuals. So we compared over 1,000 models, GLM models, and all our top models agreed on 2 top significant drivers: the number of dams and the temperature. Other drivers were also somewhat important and included electrical conductivity, distance from the sea, water velocity, where habitat depth, and water pH. So on the graph, we can see the effect of both temperature and dams, and we can observe that the presence of M. mossambica is only predicted for higher temperature and no dams. When there are other 3-4 dams, the chances of finding an eel, even at ideal temperature, is becoming really low, which makes sense because if you put more and more obstacles in their way, they are going to grow and move slowly between obstacles, and once they try to reach the top of the catchment, they're actually too big to go over obstacles they could do when they were not greater than 15 centimeters long. So our results were not all good news at all for African eel. It seems a bit all doom and glooms. However, there are issues that were out of our hands. There are very limited and fragmented data available, and no long-term or focused monitoring, as well as a lack of knowledge in ecology and biology. And worst of all, there could be issues with species identification, as eel are very difficult to ID on the field solely based on morphological characteristics. So we are working on these issues, and we already published studies on the behavior of eels and also on their genetic. So let's look at our first eel telemetry study in Africa, which was done in the Tugela and we just finished last year. We mainly looked at home range and core area, but also found out seasonal use of habitat for the different species tagged, as well as winter dormancy, which makes sense with our previous results showing the importance of temperature. So this is going to be quite interesting in designing better surveys and monitoring, but also to inform water and river management to conserve these flagship species. As species identification on the field can be an issue, we explored the use of CO1 barcoding, and we found out that this technique can differentiate the species. It's quite interesting because it's a non-lethal and accessible technique. However, you don't get 100% certainty right away on the field, as you need to go to the lab first. We're also exploring ADNA route, and from this year onward, we'll be looking at that. This could be particularly interesting as eels are notoriously difficult to catch. So in conclusion, we are concerned on a possible continued decline of the 4 African eel species. And it's not naive to think that things are going to get worse, and that bicolor and bengalensis, for instance, could disappear from our waters in the next decades. We do recommend and insist on the implementation of transboundary e-flows policy, and they must take ecological process into consideration. Fish passage solutions should also be explored. There are over 4,000 dams in South Africa and only about 60 fish passage solutions, and we don't know the efficiency of any of these solutions. So monitoring of river connectivity as well as fish and eel as in general should be conducted as much as possible, and we should keep gathering knowledge on these precious species, on their ecology and biology. Eels are, after all, flagship species for river connectivity. They are umbrella species for aquatic conservation. If you do care for them and our migratory species, you care for the life of the river. So that's it for me. Thank you so much, and we're gonna finish this presentation with a video of a longfin eel being released in the Tugela River after being measured. I'm happy to take any question. Thank you again, and hope you have a really good, lovely day.